A very good afternoon to Dr. Chi. We are from Group 2 and today we will present about the part B, which is the topic that we already choose, is method in self-pollinated plants. For the introduction, for this part B, self-pollinated plants are plants that transfer pollen from the male organs that we call as anther to the female organ stigma of the same flower or from one flower to another on the same plant. This process results in fertilization without the need for pollen from another plant. Self-pollination also ensures that offspring have a similar genetic makeup to the parent plant and maintaining genetic stability. For example, include tea plants, wheat, and rice.
Hi, my name is Laudia binti Simon. My metric number is BR23110176. I will tell you the objectives of self-pollination and methods and benefits of self-pollination. The objective of self-pollination is disease pest resistant, high yield, better quality, agronomic character, moisture, stress and salt tolerance, and wider adaptability. The methods of self-pollination is pedigree methods, which selected based on the ancestry and performance. Next, bulk method, which growing large population plants from selected parents and allow them intermate naturally. Lastly, mass selection method, which crossing a hybrid offspring with one of its parent plants. Next is the benefits of self-pollination, which is improved traits, which helps in the systematic improvement of particular traits. Next, diversity, which maintain genetic diversity in the population, which can be used for long-term adaptability. Lastly, adaptability, which effective for self-pollinated crops and can be used to improve a wide range of traits over time.
The pedigree method is a genetic evaluation technique used to trace the inheritance of specific trait or characteristic through generation. This method is commonly used in animal and plant breeding to understand how traits are passed down from one generation to the next, helping to select individuals with desirable traits for breeding purpose. In the pedigree method, a family tree or pedigree chart is created, showing the lineage of individuals and their genetic traits. Each individual is represented by a symbol, typically square for male and circle for female, and the relationship between generations are indicated by line connecting parents to offspring. Traits of interest such as disease resistance, productivity, or other desirable characteristics are recorded for each individual, allowing breeders to make informed decisions about mating or selection based on genetic history. The pedigree method helps breeders avoid inbreeding, predict the likelihood of certain traits appearing in future generations, and improve the overall genetic quality of the population.
Next, I will explain about the example of diagram for bulk method. For the year 1, in the first year, the parents plant are crossed to produce the F1 generation, which is then planted in bulk and space. For the year 2 till 5, in the second to fifth years, seed from each generation are planted at commercial seeding rates. By the 6th year, superior plants are selected through space planting, which ensures proper evaluation. Next, in 7th year, the best plants are used to establish family rows for further evaluation. For the years 8, preliminary yield trials are conducted in the 8th year to test the performance of selected lines. Then, for the years 9 to 11, advanced yield trials are carried out in years 9 to 11 to confirm the stability of the lines. And the last is, for the final steps, the best performance line is released as a new cultivar for farmer use. That's all. Thank you.
For the diagram mass selection and also the pure line selection, we can see here in mass selection a bulk of phenotypically similar plants is selected from a population. These plants may be genetically diverse, but they all share a common desired trait or set of traits. And then the process that involves, we can see, for example, is selecting a large group of plants that exhibit the desired traits, such as yield, disease resistance, or other agronomically important characteristics. For this mass selection, it leads to the development of heterogeneous cultivars, which means the resulting populations is genetically diverse. These cultivars can be registered and marketed as a single variety, even though the genetic makeup of the population is not uniform. And then, pure line selection is, on the other hand, focuses on selecting individual plants with desirable traits and then propagating them to establish genetically uniform lines. The process involves the following steps. For the first one example is selection of a single plant from the population that is often the best performing plant with the desired traits. And also this pure line selection is to create homogeneous cultivars that is genetically identical and stable over generation. Also, the line produced, for example, L1, L2, L3, Ln, that represent successive generation or individual lines that have been selected for superior traits. For the explanation about this L1, L2, L3, and also the Ln, I read from this info. For the L1, this is the first generation of offspring derived from the initial selected plant. It represents the progeny of the single parent plant after self-pollination or controlled cross-pollination. For the L2, this is the second generation, which comes from selecting the best individuals from the L1 generation and self-pollinating or crossing them. At this stage, there may be some degree or genetic uniformity, but the process of selection continues. For the L3, this is the third generation, where further selection is done to enhance uniformity and stability in the desired traits. At this point, these plants are becoming increasingly genetically uniform. And then the last one is Ln, which is represent the final generation or line. After several cycles of selection, where the plants have reached a high level of genetic uniformity and are considered pure lines. Ln is the most stable and homogeneous line, suitable for registration and commercialization as a pure cultivar. And also, once Ln is established, it can be registered and marketed as a pure line cultivar, which will produce consistent and reliable results in subsequent planting. That's all. Thank you.